Hello everyone, Dr. Ashes here. Today I want to have a brief explanation about the different methods that the pharmaceutical companies all around the world are using to create the coronavirus vaccine. There are so many companies right now are racing to create the first active and potent coronavirus vaccine. At the moment, Adelaide Lab from Australia is ready for the human trial. The CanSino from China get the permission to do the test on military, and Covaxin from India also get the permission to start the human trial on August 15. But does all of these coronavirus vaccine are following the same protocol and method? No. They all have a different methods for their own vaccine, which I'm gonna explain for you in this video. If you're interested, please follow me. The first method is a virus vaccine. In virus vaccine is the method that they whether use the weakened virus, which they make the virus go through the human or animal cell, it get mutated and it get less infectious, then they use it as a vaccine, or they use the several chemical to inactivate the virus, then they use that. But what is the pros and cons of these two methods? The upside of this method, the good thing about this method is that this has been established for a long time, so the capacity for mass production is already there, so it's very easy to reach the mass production. But for downside, it required long-term tests. Because they're introducing the real virus inside our body, they need to make sure that this virus doesn't actually infect the body. So they need maybe 4 years, 5 years, 6 years. So it's not a method that it can get the answer in 1 year, 2 years. Then the second method is using viral vector vaccine. In this method, it's involved genetically engineering the virus, and at the same time, they're using two different methods. One is replicating the viral vector vaccine, which the virus they introduce to the body, it can replicate itself, or they're using non-replicating viral vector vaccine. In this method, they only introduce the virus to the body as a vaccine, the weakened one, but it cannot replicate itself. So what is the pros and cons for these two methods? The upside is there was a successful trial on the Ebola virus vaccine, which came out successfully. But downside is any sort of pre-immunity that the people have can destroy the effectiveness of this virus, of this vaccine. Therefore, it can be less effective for some people, and some other need to get a booster shot in the year. So it's not one shot and you will be immune for the rest of your life. So next method is using protein-based vaccine. In this method, they mostly work with the spike of the spike protein from the virus, which is involved in attaching to the ACE2 receptors and infecting our body. For this matter, whether they use the protein subunit vaccine, which is right now 28 different companies are working on that. This approach involves manufacturing the massive supplies of the spike protein and introduce it to the body as a vaccine. And the next method is a virus-like particle vaccine, which in this part they use a fragment of the protein of the vaccine, which mimicking the coronavirus outer coat will be used to introduce to our body, so to start the signaling to our body that our body is invaded, so producing antibody and get immunity. The upside and downside of this is that first of all, the vaccine are much safer is a good part of it, because it's not involved the whole virus in introduction to our body. It's just part of the protein. It's much safer. But we had test the SARS vaccine with the same protocol, and it was successful on monkeys. But downside is they have been never tested on the human, so we have no record of the long-term positive side effect or the effect that this virus can have on human. Therefore, it's very risky to use this protocol. The last method is a nucleic acid vaccine. In nucleic acid vaccine, they whether using DNA or RNA, which they inject this material to the body, and this genetic material will encode the virus spike protein, which body will recognize it as an invader and produce the right antibody to fight it. For DNA, they need to pores are created in the cell membranes to increase the uptake of the DNA in this method called electroporation. And for RNA, they need to coat the RNA with a lipid that it can help it to enter the human cell and also copies the virus protein. The benefit of this method is that nucleic acid-based vaccines are often safer and have a potential to be developed quickly, but the downside is there has been no test of such vaccine on human before, so they need to be very cautious about it. So these are the methods that are being using currently to create the coronavirus vaccine. I hope you enjoyed this video. If you found it good, please share and subscribe. And till next time, please stay safe and